So this is the third year of the summer science course for the Regent Science Scholars Program. And um, so I'm a scientist, so I'm comfortable doing experiments. And so I think that anytime you design a course, it's an experiment, right? So we have ideas and we try them out and then we see how it went. We see how students grew during that summer's work, but we also then see how it's prepared them or not prepared them as well for the coming year. And I think we felt that after the first couple of years, we were hitting on some points and not hitting on others. I think we were successfully bringing students into science classrooms. We were giving them a lot of hands-on experiences, which many of them didn't get in high school, for actually doing experiments and rather sophisticated experiments. And we were clearly helping to convey a sense of the rigor and the high level of expectations in coursework. But part of what we're about in college as well is that we're not just about teaching the facts of science, we're about science as an intellectual endeavor and about being creative and thoughtful around it. And we're also very much about science being about community. I think a lot of students get out of high school in general thinking about any intellectual work as solo work, right? Because you write your own papers and you take your own tests. But science as a human endeavor is very much about community. And so the nature of the discipline and what we're hoping for the students to be able to form these strong supportive communities meshes very well. And so I wanted to redesign it this summer to do a better job of being about science, real science, not just science content. And so the opportunity for that has an interesting and slightly bizarre history. But we were wine tasting at a winery in Virginia, the Glen Manor Vineyards. And they had barrel tastings. And the vintner, a man named Jeff White, was explaining that in this particular tasting, what he wanted us to taste were what he was calling feral wines. Feral, like wild wines. And the reason was that when most winemakers do the fermentation process, they do it like we think about making bread, right? So they kind of get all the ingredients together, in this case grape juice, and then they add yeast. So it's not quite your like packet of Fleischmann's yeast, but it is the winemaker's equivalent. People around the world use the same strains of yeast to do the work of breaking down sugars and making alcohol from that. So, but he was trying a technique that others have also played with. So we had gone out to this vineyard for barrel tasting of wines, and lots of vintners do this. And it's based on different, rainfall conditions or grapes or whatever from the previous year. But in this particular case, the distinction that Jeff was making was between wine that was being fermented using commercial yeast. So something that's predictable. Every year he adds the same thing, and it should be doing fermentation the exact same way of breaking down sugars into alcohol. But what he was creating was something called feral wines. And the feral part of the wine was saying that the wildness was from the microbes who were going to do that process of fermentation. And so like most vintners, he doesn't wash his grapes or things like that. They pick them carefully, they bring them in, they sort through everything, and then they crush it, which means that if the surface of the grapes have any bacteria or fungus of their own from the wild environment, that's going to be what's doing the fermentation. And this seems to freak everybody out, but it's actually healthy that basically everything in the world is coated in microbes. So people are coated in microbes and grapes are coated in microbes, and that's what he was using. And the taste difference was dramatic, and it was very, I guess from a wine perspective, it was lovely. But he was saying that he was a little frustrated because big wineries have the ability to actually understand scientifically which microbes were part of that community and were shaping the flavor profile of the wine. But although he's an award-winning vintner, it's a very small family winery. And he just said he wished that he could better understand this. This is how people are always like, how do you come up with your ideas when you teach? So this is how I come up with my ideas when I teach. So I was like, wow, Jeff, I can be helpful. And was deeply embarrassing to everybody around me at that moment. But we started talking, and he said, "You have a lab that does this?" And I said, "Yeah, I'm going to have about 30 students this summer working with me." And so helping Jeff identify the community of microbes that were both shaping the plant growth, so the grape growth itself, and then the fermentation process became the research project. And so then everything else this summer happened, in the sense, in service of that research project. So that was the work that we did most of the days in the lab. And the content, what we talked about, we just kept pivoting back to talk about why you need to talk about basic ecosystem biology and population and community structures. That's something we would talk about anyway, but we talked about that in service of his project. We talk always about diversity of life, but diversity of life is actually largely diversity of microbes, because microbes are most of life forms on Earth. So we did it in that service. We always talk about cellular energy processes, but that's what fermentation is for a cell. It's how it gets energy. And so everything kind of dialed back to that. And the work that we did in the lab was we went out and we collected 16 samples from his vineyard and analyzed the microbiome communities that were within those samples. Exactly the project that we did, we had the students design their first week in class. They were surprised at being handed that responsibility. A little dismayed that we needed to agree, but that's how teamwork works. You have to agree on stuff. So we spent many hours that first week debating it. The phrase, "What would best help Jeff?" "What would Jeff want us to do?" Became catchphrases. And I think one of the things that was captivating about the project was how quickly the students embraced, were enthusiastic about this idea that it mattered, that they weren't just doing hands-on stuff, but that their data would matter to somebody. And so with everything happened, like, well, I don't know, how should we plate these out on Petri dishes? I don't know what would help Jeff. How do we parse the data in the end when we were all said and done? Well, I don't know, what form would it help Jeff most to see it in? Kind of became the driving logic. And so we pursued two very different things. We went old-fashioned microbiology, and they plated stuff out on Petri dishes, and we looked at diversity by visually, by eye, by microscope. We also looked biochemically at kind of some of their different properties. But then we also went super modern and high tech. So one of the huge cutting edge fields in science right now is a field called metagenomics, where you try to understand from DNA sequences all the members that are present in a community. And we've learned from those sorts of things that life is about 10 times more diverse than we ever thought it was. And so the students did this work. This is work, we read primary scientific literature of other groups doing these exact sorts of studies. The only downside, which is an upside, because the glass is always half full, is that we didn't finish the work this summer. And the students were upset that we weren't going to finish. But so that's okay, because they're now a whole new research team, and we're doing research through the year coming up. So I have freshmen coming in who've never done research before in their lives, who made meaningful work happen this summer, but who are sufficiently committed that we're keeping it going as a research project with a goal of publishing it. But we owe all of this back to our partnership with Jeff. So at the end of the research unit, we went out to the Glen Manor Winery, and Jeff gave us a tour of the vineyards. And one of the things I should still get from my students, because they'd each been responsible for different patches within the vineyard for their analysis of the microbes. We had students taking pictures of dirt underneath plants, because it was their dirt, and like grapes and leaves and things like that. And Jeff took several hours to talk with all of them. My students had prepared a whole series of presentations, so we divided the project out into all these different groups, and they each claimed ownership of trying to explain a piece of the project. But that required them not just to explain their own work, but that of their peers also, which is, again, how scientists explain things. And And so Jeff and his nephew and niece also were present at all of this and helping out. Listened to it. I got an email back from Jeff afterwards. I mean, I wrote him thanking him. He wrote me back thanking me. And his only concern was if we were going to keep the project going so that he could continue to hear more of the data, and that he already had more ideas for us for next summer when the next cohort of students come on board. And my students this year have already asked not just if they can keep the research going, but can they come back to be TAs in the course next summer, because they want to help the next generation get going on this research. And that's what science looks like. That was cool.